Good evening, class. Our first lecture for tonight is about the history of molecular biology. At the end of this chapter, you are expected to outline the significant discoveries in the development of molecular biology as a science, identify the Nobel laureates and other scientists with their respective contributions to molecular biology, point out the significant relationship of earlier discoveries and findings to recent methods in molecular biology, and create a timeline of the milestones in the history of molecular biology. Just for an outline of the discussion, first we will discuss about the origin of molecular biology. We will discuss about the transforming principle, DNA as the genetic material, Watson and Crick's model of the DNA. We will discuss the semi-conservative DNA replication, the central dogma of molecular biology, and notable contributions to the development of studies in molecular biology. Originating in the 1930s and 1940s, molecular biology emerged as a remarkable convergence of diverse scientific disciplines, including biochemistry, genetics, microbiology, physics, structural and colloid chemistry, and virology. This interdisciplinary synergy propelled molecular biology into the scientific spotlight, with its momentum skyrocketing in the 1950s and 1960s as it became institutionalized. But what exactly is molecular biology? At its core, it is the study of biomolecules and the intricate molecular mechanisms operating within living organisms. While biochemistry also delves into these realms, molecular biology distinctively focuses on the structure and function of the gene. From the molecular nature of genes to gene replication, mutation, repair, and expression, molecular biology offers a unique lens through which we decipher the fundamental processes of life. The term molecular biology itself was coined by Sir Warren Weaver in 1938, marking the dawn of a new scientific frontier. He envisioned a realm where cutting edge techniques would unveil the mysteries of cellular processes at the molecular level, a vision that continues to drive our exploration today. Now, our journey into the origin of molecular biology wouldn't be complete without acknowledging the pivotal contributions of pioneers like Gregor Mendel, whose work with garden peas laid the groundwork for understanding hereditary patterns, and Friedrich Meisker, who first isolated nucleic acids in 1869. These early milestones paved the way for groundbreaking discoveries, culminating in the elucidation of DNA's biochemical structure in 1953 by Watson and Crick, a watershed moment that forever altered our understanding of genetics. Sir Gregor Mendel's contribution to the field of genetics is foundational and widely acknowledged. Through his meticulous experiments with pea plants in the mid 19th century, Mendel discovered the basic principles of heredity. His work demonstrated the existence of discrete hereditary units, now known as genes, and elucidated the patterns of inheritance for traits such as flower color, seed shape, and plant height. Mendel's findings, published in 1866 as Experiments on Plant Hybridization, laid the groundwork for modern genetics. Despite the initial lack of recognition, his work was rediscovered at the turn of the 20th century, leading to the establishment of Mendelian genetics as a cornerstone of biological science. Now, one of Mendel's most famous experiments involved crossbreeding through breeding yellow and green pea plants. He found that in the first generation, all offspring had yellow seeds, suggesting dominance of the yellow trait, as shown here. However, in the next generation, the trait reappeared at a ratio of 1 green to 3 yellow. To explain this phenomenon, Mendel introduced the concepts of recessive and dominant traits. In this example, the green trait, although seemingly absent in the first generation, was actually recessive to the dominant yellow trait. Mendel's groundbreaking work was published in 1866, where he proposed the existence of factors, now known as genes, that determine hereditary traits in a predictable manner. Sir Frederick Maesker, a Swiss biologist, made a significant contribution to the field of molecular biology by discovering nucleic acids in 1869. While working with the white blood cells, Maesker isolated a new substance that he termed nuclein. This substance, later identified as nucleic acids, proved to be a crucial component of cells and played a fundamental role in genetic inheritance. Maesker's discovery laid the groundwork for understanding the molecular basis of heredity and paved the way for future research into the structure and function of DNA and RNA. Watson and Crick are renowned for their groundbreaking contribution to molecular biology, the elucidation of the double structure of the DNA. In 1953, they proposed the double helix model of DNA, which revealed the molecule's elegant and intricate structure. Watson and Crick's work laid the foundation for modern molecular genetics and revolutionized our understanding of inheritance and evolution. Their discovery earned them the Nobel Prize in Physiology or Medicine in 1962, cementing their place as pioneers in the field of molecular biology. Frederick Griffith was a British bacteriologist whose focus was the epidemiology and pathology of bacterial pneumonia. In January 1928, he reported what is now known as Griffith's experiment, the first widely accepted demonstrations of bacterial transformation, whereby a bacterium distinctly changes its form and function. By injecting mice with different strains of Streptococcus pneumoniae bacteria, he observed a phenomenon known as transformation. When he combined heat-killed virulent bacteria with live non-virulent bacteria, the once harmless strain transformed into a deadly pathogen. Avery, MacLeod, and McCarthy meticulously dissected Griffith's findings, employing a series of enzyme treatments to pinpoint the elusive transforming principle. Through their meticulous experiments, they discovered that it was not proteins or RNA, but rather a molecule called deoxyribonucleic acid or DNA. This held the key to hereditary transmission. The revelation of DNA as the genetic material. Imagine a world where the blueprint of life itself was shrouded in mystery, where the very essence of heredity remained an enigma. It was in this backdrop of scientific curiosity and fervor that Alfred Hershey and Martha Chase embarked on a groundbreaking quest that would forever change our understanding of genetics. In 1952, Hershey and Chase embarked on a series of experiments that would unravel the fundamental fabric of life. They turned their attention to bacteriophages, those enigmatic viruses that infect and replicate within bacterial hosts. These phages, comprised of a protein coat and DNA, held the key to unlocking the secrets of heredity. With meticulous precision, Hershey and Chase employed radioactive isotopes, namely phosphorus 32 and sulfur 35, to selectively label the DNA and proteins of the phages. Upon infecting bacterial hosts, the DNA, not the proteins, was found to be internalized within the host cells, propelling the production of phage progeny. This seminal discovery unequivocally established DNA as the bearer of genetic information, setting the stage for a revolution in molecular biology. But the elucidation of DNA's role didn't end there. In 1953, James Watson and Francis Crick unveiled the iconic double helical structure of DNA, a feat that would earn them a place in the annals of scientific history. Their model, depicted in their legendary paper in Nature, revealed DNA's elegant architecture, two intertwining helices with sugars and phosphates forming the backbone and bases nestled snugly within. Watson and Crick's model not only elucidated the physical structure of DNA, but also hinted at its profound functional significance. The specific pairing of adenine with thymine and guanine with cytosine hinted at a mechanism for faithful replication, which is a mechanism that would lay the foundation for the perpetuation of genetic information from one generation to the next. From the groundbreaking work of Watson and Crick to the unsung heroes like Rosalind Franklin and Raymond Gosling, from the groundbreaking work of Watson and Crick to the unsung heroes like Rosalind Franklin and Raymond Gosling, the road to unlocking the mysteries of DNA was paved with the sweat and toil of countless scientific minds. It was their relentless pursuit of truth that brought forth the revolutionary technologies we now take for granted: sequencing, cloning, gene editing, and beyond. But the story doesn't end there. The marriage of biology and technology gave birth to a new era of exploration, one where data and algorithms converge to unravel the complexities of life itself. Enter systems biology, functional genomics, microarray, and bioinformatics, fields that have reshaped our understanding of genetics and paved the way for groundbreaking discoveries. Rosalind Franklin's famous photograph, known as Photograph 51, this provided crucial insights into the structure of DNA. By recording fiber diffraction from DNA fibers, this is the DNA fiber, placed in an X-ray beam, Franklin observed a distinctive X-shaped pattern on the film. This pattern indicated the helical structure of DNA with characteristic spacing and features. The first thing we learned from this photograph is that DNA is in fact helical. This characteristic X pattern is what one observes when one places a helix in the path of the X-rays. Now, the second thing we learn from the photograph comes from the position of this dark spot over here. From the distance, from the center to the dark spot, that tells us that the fundamental repeats of the helix are 3.4 angstroms apart, or the DNA helix is a 3.4 angstrom rise per base pair. Now, nakikita niyo na class tong blue line here. So the spacing between these sets of spots here, known as the layer lines, gives us a pitch of the helix, and the pitch here is 34 angstroms. So the pitch here is the distance going vertically here that the helix repeats itself. And now if you take the pitch of 34 angstroms and divide by the rise per base pair of 3.4, that tells us that there are 10 base pairs per turn in the helix. Now, if we look closely at these layer lines, you actually see that there are missing spots on this fourth layer line. So class, may missing na spots dito kasi dapat 'di ba itim to. But in this case, pale siya or white. So, ano ibig sabihin niyan? The missing line tells us that there's more than one strand wound around one another, and that those strands are not symmetrically placed, so they're asymmetric. Then that was the clue that there's a major and minor grooves in the DNA. Dito class, ito yung mga grooves na tinatawag. Finally, the spacing between the spot gives the helix radius. Ito class, yung distance between dito sa mga spots na to, that gives us the radius. In this case, ang na-measure nilang radius is 10 angstroms. So if you will get the diameter, i-multiply mo lang by 2, then we get the diameter of the helix of around 20 angstroms, which is really exactly the diameter of DNA. So you can so yan, ma-estimate nyo na class yung measurements or dimensions ng DNA from there. Next, Chargaff's experiments revolutionized our understanding of DNA's structure and composition, laying the groundwork for Watson and Crick's iconic double helix model. In his seminal work, Chargaff meticulously analyzed the nucleotide composition of DNA from various species. What he uncovered shook the scientific community to its core. He revealed that while the sequences of nucleotides varied between species, there was a remarkable consistency in the ratios of certain base pairs. This consistency birthed what we now know as Chargaff's rule, a foundational principle that set the stage for further discoveries in genetics. But what exactly is Chargaff's rule? It's the elegant observation that the amount of adenine, itong A class, is always roughly equivalent to the amount of thymine. And likewise, the quantity of guanine mirrors that of cytosine. Now, this symmetry, this delicate balance hinted at a deeper order within the genetic code. Chargaff's insights didn't just stop there. His work provided crucial evidence supporting the complementary nature of DNA strands, a concept central to Watson and Crick's revolutionary double helical model. So, what is the semi-conservative DNA replication? Messelson and Stahl's groundbreaking experiment provided compelling evidence for the semi-conservative nature of DNA replication. This model proposed that during replication, the old DNA strands separate, serving as templates for the synthesis of new DNA strands. Class, notice here yung dark blue na strand. That is the parent or original strand. And then this red here, this is the nascent or the daughter strand. So each DNA replicated is a hybrid of a new and adult and an old strand. That's why we call this the semi-conservative nature of DNA replication. Okay. Now, as a result, each round of replication yields two double helices, each a hybrid of one old strand bound to a newly synthesized strand. This elegant process ensures that the faithful transmission of genetic information from one generation to the next. So class, ito yung reading assignment. Table 3, notable contributions to the field of molecular biology by prominent scientists and Nobel laureates.